Good afternoon, everyone. I hope you had a good morning. Can I just check that everyone can hear me and see my slides? Yep, all okay, great. Okay, I'm going to be quick here. So thanks to GYSS and for asking us to do this engagement session. So we're coming to you from the Mechanobiology Institute. We're hosted at NUS, and we're one of the a research center of excellence, one of 7 currently in Singapore. So you can see that 4 of them are hosted at NUS, and I think you've heard from some of our colleagues in these RCEs over the past few days. And we also have some other RCEs at NTU as well. So yeah, we're the Mechanobiology Institute here, and what do we do? Well, our aim is to develop the world's leading center for mechanobiology research and education, try and find ways to translate this, and also lead the way in sort of education in mechanobiology. Next question obviously would be, well, what is mechanobiology? So on the broad scale, it's what we do here at MBI is to sort of interrogate and examine biological systems from a physical and mechanical perspective. So one example would be protein folding. And, that is, while it's a biological or biochemical process, there's also physics and mechanics in that. The same is true for DNA replication, what you need to sort of split 2 parts of DNA and then put them back together. So what we have lined for you today or this afternoon is first we have a talk from Dr. Andrew Holly. He's a principal investigator here at MBI, and he's an NRF fellow as well. And he's going to talk a little bit about his research in confinement mechanobiology and his journey to Singapore to become an independent investigator. And then we'll also have 3 PhD students from the MBI graduate program, Malia, Jashan Singh, and Martin Kiwanuka, here to give you some just experience of what it is about mechanobiology, MBI, and Singapore that attracted them to come and work in this interdisciplinary field. Okay, with that, I'm going to stop sharing this slide and let's see. And I'll be putting up the slides for Andrew Holly.
Okay, hello everyone. Thanks for joining us today and giving me the opportunity to talk a little bit about what I've called here as a migration story. So not just cells, but also how I ended up in Singapore and the type of research that we actually do. So just as a little background, about where I've been on my academic journey. Andrew used the word journey earlier, which I think is really appropriate because the research that we do also has to do with the journey of cells. But for my entire research journey, I've been really interested in how single cells sense their physical surroundings and then change their behavior accordingly. So I'm from the US originally. I did my PhD in San Diego on stem cell mechanobiology, so how focal adhesions could sense their environment. Then I moved to Germany, where I did a postdoc in the Max Planck Institute. And throughout the whole process, one of the things that I've really come to enjoy about the academic lifestyle, let's say, and research is it's truly international, right? And a lot of the collaborative work that I've done over the course of my career has involved international collaborations, including with Australia, with Germany, and places all around the world. So during my PhD, which I started way back now in 2009, I was really interested in how stem cells might feel their environment. And one of the ways that stem cells might be able to feel their environment is through these molecular strain gauges, where the more force that's applied to a certain protein that connects the cell to its extracellular matrix, the more unfolding we have. And cells can sense this unfolding, right? That's what we see in this video. So the protein that I was particularly interested in was vinculin, which we identified through a screen as playing a major role in stiffness-based mechanosensitive stem cell differentiation. So by reducing the amount of vinculin inside cells, we found the cells were much less able to sense their stiffness of their surroundings and then differentiate accordingly. And we had done some bioinformatics-based work to try to identify how this actually happens. And ultimately, we found that this works through the MAP kinase pathway. So then when I moved to Germany, I was interested in moving into a different type of science. This is kind of a beauty of doing graduate work and postdoc work, is that you're not limited to just what you did your PhD work in. I became really obsessed with this idea of how cancer cells might squeeze into very tight spaces, because cancer cells have to migrate from their home tumor to a distant site of metastasis. And if we can interrupt this process, we might be able to actually prevent metastasis, which is what is so dangerous and deadly about cancer in general. And we know from intravital microscopy that cancer cells can squeeze into very tight spaces. So as a bioengineer, one of my goals was to replicate this type of environment, but not in the body, in the lab, right? And so we've done photolithography and PDMS-based replica molding to actually build microchannels that our cells can migrate through. And this is a very dynamic process, as we see in these videos. And how these cancer cells move in tight spaces actually depends a lot on how tight the space actually is, right? And this matches what we would envision for our own daily life as we migrate from point A to point B. If we're walking through a very wide hallway, we walk very comfortably. But if it's a very, very tight space where we have to squeeze, we might move a little bit differently. And this is such a well-analogized process that it's actually referred to as chimneying, which comes from the rock climbing community, where climbers don't actually have to grab on tightly to anything. They can just push outwards if they're in a space that provides enough confinement. So the new directions that I've started since coming to Singapore, since joining the Mechanobiology Institute in the past 2 years, is the idea of focusing on biology and mechanobiology, not just in static environments, right? Of course, we have to understand what the bone marrow looks like in order to understand bone marrow cells. We have to study the characteristics of lymph nodes to study the immune cells that are found inside them. But I want to move to this new idea that maybe the migration during the journey that cells take, they're influenced by the mechanics of the spaces that they actually move through. We have a saying in the US, I'm sure there's other ways of saying this around the world, but people like to say it's the journey, not the destination, right? But what we kind of think is maybe for cells, it's the journey and the destination. So we should be studying both of these different phenomena, especially because the types of environments that cells move through can be so wildly different, right? So one of the first things that we're interested in my lab is understanding the interstitium, characterizing it. These are fluid-filled highways that migrating cells move through, and when they migrate through these spaces, they're exposed to different levels of compression. They're squeezed in different ways. So we know that there are characterizing these spaces in the body is a little bit difficult, but recent methods have allowed us to understand this a little bit better. We know that bone marrow cells can migrate long distances in vivo based on experiments with mice that you put green fluorescent positive cells from the bone marrow into mice that don't have any green fluorescent positive cells. And then later, weeks later, you can find these green fluorescent cells all over the body, which implies that they start in the marrow and then migrate elsewhere. But we still don't know too much about what these spaces actually look like. And thus, we don't know what type of confinements and compression cells are exposed to. So our first goal in my group is to measure these highways that cells migrate through in both healthy and diseased tissues. And there are many interesting bioengineering-based ways that we can do that, but one of the ways that we use is called tissue clearing, where we can actually take an organ that we wouldn't be able to image inside, apply different chemical treatments that allow light to penetrate and pass through these organs, and thus, using this technique in conjunction with confocal microscopy, we can make direct 3D observations of these spaces. Now, once we've imaged these spaces, once we know kind of what they look like, the next goal is to actually copy it, right? To build confinement-based devices that mimic these spaces and control multiple variables, whether it's the width of these channels that we make, the types of patterns the cells are exposed to, the nanoscale topography, et cetera. Lots of things that we can actually look at. We've actually have a proof of principle essentially now that if you look at stem cells, so not cancer cells, but stem cells that would migrate from the bone marrow, these cells are actually able to migrate in these very tight spaces, even though they're much different cells from stem cells. And in fact, what we find is that by controlling just the confinement degree, right, from 3 microns to 10 microns, we can get significant changes in the amount of bone differentiation these cells will actually undergo. So cells that migrate through very, very tight spaces are much more likely to differentiate towards bone-like cells. Now, what's nice about this is that if we can control differentiation based just on a physical property and based just on migration, then we might be able to scale this up to assemble tissue structures based on this differentiation. So many of the tissues in our body are highly stratified. In this particular picture, we're looking at retinal tissue. You see there's many different layers of very specific differentiated cell types. What our goal is to actually put patterns of channels that can stimulate different types of differentiation in very specific spatial arrangements that allow us to actually build these interfaces that we would find in the body that otherwise would be very hard to control just through chemical differentiation. And a very good simple example of this is the interface between the muscle and the tendon. So these are muscle cells and tendon cells that come close to each other. They form this interface, this gradient. But this particular interface has been very challenging to engineer and to study in the lab. So ultimately, overall, my lab is called the Confinement Mechanobiology Lab, and this is our general approach. We know that stem cells in the body will migrate through various diverse spaces in order to achieve their goals in the body, right? To differentiate into pure cell populations, to undergo an immune response, and to form patterns of tissue during development and wound repair. Our goal in the lab is to take stem cells, apply them to different types of diverse confinements, mostly through microchannels, and then recapitulate what happens in the body, either by making pure cell populations, studying immune response, or patterning tissues, but then go beyond this to do things that we couldn't do in the body, like mechanically priming cell populations, or the development of commercial devices that allows other labs to actually impose this type of confinement. Ultimately, what I want you to kind of take home is that with this type of confinement mechanobiology and how cells are responding to their environment, it's probably the journey and the destination that's very important. So with the last couple minutes of my little presentation, I want to quickly reiterate some of what Andrew mentioned about the Mechanobiology Institute, which is where my lab does its work at NUS. There are four main research themes, right? Mechanobiology occurs at many different scales, from molecular mechanobiology to cellular mechanobiology, all the way up to multicellular mechanobiology. We're looking at organs and development. And my lab kind of fills this cellular mechanobiology area, but it's very nice to be able to communicate and work together with other researchers that are looking at different scales and different levels of mechanobiology. One thing I want to emphasize, because this is the global young student symposium, is that our community at MBI is very global, right? We've got, I believe, researchers representing 6 of the 7 continents at this point. And so I think this is a really exciting environment, both at MBI and within Singapore, to be exposed to researchers from all around the world. Our presence is also very global. So this is just a quick example of some of the types of conferences that we actually send students to. So our research is definitely highly interdisciplinary, highly international. So if you're an undergrad who's considering a career in research, I would encourage you to join us. We have a summer undergraduate training program that we haven't hit the deadline yet, so you can certainly still apply and come here to do summer research. If you're considering a PhD, we offer PhD scholarships. This program is also open for applications right now. If you're thinking about doing a postdoc, if you just finished your PhD, many labs at the MBI, including my own lab, are looking for postdocs right now, so please join us. And finally, if you're finishing up your postdoc, even at the faculty level, we're open to new people as well. So overall, thank you for your attention today. Thank you for letting us talk a little bit about mechanobiology, which is a bit of a obscure subfield, let's say, but we think is highly important for many different levels of biology research. And with that, I'd be happy to take any questions or pass over to our students. Okay, no questions. Great.
Okay, I guess we can move on to a little bit of a sharing session with our PhD students. If you do have any more questions, do drop them in the chat and we'll do our best to answer them. So I think, let's see, I'm not sharing anything there. Yes, I'm joined here by 3 of our MBI PhD students here. Meng Yue, who's joining us from abroad at the moment, Jashan Singh, who's in the room with me, and Martin Kiwanuka here. So I thought we'd just ask them a few questions. And again, if there's anyone in the audience who is interested in learning more about mechanobiology or the MBI PhD program, then please let us know and what questions you have, and we'll try to answer those. So I guess, well, let's see. Let's start with here. It's just like, let me start with Malia, since she's in the other room right there. So it's like, so could you talk a bit about your sort of background here as an undergraduate, and I guess in the spirit of what Andrew was talking about earlier, your journey to MBI?
Okay, thank you, Andrew. So, hi guys, I'm Malia from MBI, and currently I'm a third year PhD student. So I'm very happy to join today's interaction session and share my own experience. So previous, I have got my undergraduate degree and also master degree in China. So I majored in biology for my undergraduate and moved to Chinese Academy of Sciences to study on the say elegance gap junction, which is a type of the am I reconnected? Yeah, so it's kind of type of the cell-cell junction in the elegance. So at that moment, I have been attracted by the beauty for the biology. However, my most study before my PhD are more focused on the biology, pure biology. I do not have in different background or like engineering to develop different ideas. So at that moment, I realized to learn more or to find more, explore the biology beauty, I may need to learn some different knowledges. So at that moment, I learned about mechanobiology, since it can give me a more chance to learn how the different environment can infect the cells or infect the organism. So at that moment, I choose to join MBI to explore my future research journey.
Well, actually, how about I pass this over to Jashan? It's like, how did you end up here?
Hello, everyone. So my name is Jashan. I'm also a third year PhD student here at MBI. So my undergrad was from the Indian Institute of Technology, Roorkee, where I did life sciences and specialized in nanotechnology as well. So that journey took me around working with graphene and aptamers and aftersensors, and that involved with a little bit background in mechanical designing of products. As an intern, I thought maybe I can fuse both mechano products and also biology. So I thought mechanobiology would be the best fit for me, and it was. So mechanobiology, right now I am working with biomaterials and working on cardiac aging, which is, as you know, aging and longevity is very much explored at this, it's a very high field at the moment to be explored. So yeah, so I am currently exploring cardiac aging on different biomaterials.
So I think even from that, you can see we have like someone coming to mechanobiology from a biological background, and then someone coming more from a sort of engineering and nanomaterials. So I think one of the things that we're trying to emphasize here is that, well, even at the principal investigator level, we have professors with background in physics, mathematics, engineering, even some in sort of biomedical sciences and medicine. So mechanobiology is a very diverse, broad field, and actually lots of the questions that we are trying to answer and lots of biological questions actually have a mechanical and sort of physical underpinning. So let's see, there's one biologist or a biology background, one engineering, and let's we can ask Martin to see what's his one.
Hi everyone, my name is Martin. I'm from Uganda. So I did my bachelor's degree in biomedical engineering at Malia University in Uganda. And due to my interest in diagnostics, I moved to University of Cape Town in South Africa, where I joined Professor Thomas France and did my research in cancer mechanobiology. And during this master's research, that's when I got to know about MBI and NUS in general, because most of the papers I was reading for my research were actually from researchers at MBI. So I picked interest in MBI, and I tried to find out more about MBI, NUS, and Singapore. and actually that's why I ended up coming to Singapore and MBI for my PhD. In my PhD, I'm also using biomaterials to build tumor models to mimic stiffness, matrix stiffness of tumors. And I'm using these models to study a lot of matrix stiffness in cancer therapies. And I'm using breast cancer and liver cancer as my models. And the kind of treatments I'm looking at is chemotherapy and immunotherapy. And I'm trying to see how these treatments are affected by the various stiffness of the tumors.
Okay, so I think that's a little introduction to the three PhD students that we have here. And yes, I think that Martin spoke a little bit about his project that he's working on, sort of the stiffness and ECM. So Jashan, I think you said a little bit about yours. I know you do a similar thing working on sort of the extracellular matrix, but could you share a little bit about, like, I guess in which ways it's similar and some is different, like because you're looking at the heart?
Yes, so that's a good question. So I'm working on a cardiac aging model. So to find out how heart so the heart stiffens as you age, and I'm trying to mimic that in vitro. And also we have an animal model that I can get primary cells from, primary cardiac cells from at different age points of the animal, and then we try to replicate what is physiologically relevant in the heart, and we then go for different treatments on the cells to see what can be beneficial, or it can actually either halt aging or, in our case, reverse aging, but that's still a long shot, and we're trying to find out what is possible in order to reverse aging or halt aging.
Yeah, I think that's one of the sort of subjects where mechanobiology is really sort of advancing forwards in terms of aging, because it happens to everyone, and in some core it's very mechanical, right? Your body starts to break down, so the organs don't work as well. As you said in the heart there, it doesn't pump the blood around the body as the same efficiency as power as when you're younger. I know as I'm getting older, muscles started to weaken a little bit, so you need to do a little bit more force bearing and force loading exercises just to keep everything in sort of tip-top shape. So yeah, mechanobiology, aging, it's definitely a very big field that we and other researchers are moving into. So I think a little bit differently, yes, your project is, if I'm correctly, looking at sort of structure and function of was it cell adhesions and those components? Is that correct? So Mengyu, I think that your PhD program is your project's a little bit different. Is that correct? You're looking at
my project is quite different with Jashan. So I'm starting the cell junction. So because my previous study we like the gap junction formation and the molecular mechanism in C. elegans. So after that, I realized Tony's group, and we are doing like the study on how cell is connected and how cells is communicate between each other, and how it can sensing the environment and doing the mechanical transduction role. So my project is starting like the proteins which form the cell junction structure called catenins. So I'm recently studying like how catenins can affecting the cell like bonding the connections. So that's my current project, and we are still exploring how different cell junction may communicate each other.
Sorry, that's great. So yeah, again, we have a very wide range of projects which are going on here, and I think at the moment we have about 40 to 50 graduate students in different stages of their projects, again working from all things related from sort of the molecular mechanobiology of like focal adhesions or cell or cell matrix adhesions, all the way up to like larger model scales about organ development and small organism development there. Okay, so I thought that's a little bit about the science of all our PhD students and what they're working on here. So yes, I guess it's been covered a little bit, but just like maybe I'd like to ask someone else, like, why did you choose to join MBI? There's lots of PhD projects around here. It's like, what was it about MBI that made you interested to come here? Let's start with Jashan. Sorry.
What made me join MBI? I think I already covered because of the interest in my background, doing both mechanical engineering and nanotechnology. I wanted to make the best of both worlds. As to why I chose the lab I am in right now, it's more to do with also with the background and also how the research can be taken forward. I really enjoyed enjoy the projects that were presented to me, and this was one of the areas I really wanted to go into. That's why. So that's why MBI in general. If you ask me why Singapore, Singapore is very close to India, and it's just 5 hours away, and the weather is always constant, which can be a plus or minus, but it's a very nice country to live in. Yeah, that's why I vote MBI.
Yeah, so Martin, I think you are definitely our first PhD student from Uganda. So it's like,
Yeah, I'm the first PhD student for Uganda and I think Africa.
I think so too, yeah.
Yeah, so like I briefly said during my first I got to know about MBI when I was doing my master's research. My master's research was in cancer mechanobiology. And I got to know that MBI is one of the leading institutes in this area of research. And when I got to look at the different research topics that are being handled at MBI, I saw that it would give me a chance to merge biology or medicine and engineering, because my bachelor's background was in engineering. But at my master's, I didn't do much of engineering because I was venturing into a new research area, and because of that time that we have for master's, I didn't get much time to incorporate in the engineering. So I thought that if I join MBI with the kind of interdisciplinary research that is being done here, I'll have an opportunity to match the two and get to build more on my understanding of mechanobiology from both biology and engineering aspects. So when I came to MBI, I chose to join. I rotated in a few labs, like in 3 labs, and out of this, I chose to join Professor Jennifer Young, whose research is in he's looking at soft nano environment within the body, and is applying biomaterials. So I thought I would have a chance of doing research in biomaterials, which sort of brings out the engineering aspect, and then biology by studying how if I use biomaterials to make models, like I'll be using these models to understand how cells behave in this kind of environment, and also with different treatments, because I'm interested in cancer, with different treatments for cancer, I can understand how this environment that I'm building are affecting the treatment, and so that there's a way we can maybe identify more targets for cancer treatment, or ways of diagnosing cancer at the early stage, because we know that, especially where I come from in Africa, one of the reasons why people are dying of cancer is because it's tested later in the development of cancer, because people take long to go to hospitals and take long to know that actually what they have might be cancer. So this is giving me an opportunity of trying to show that there is a way we can diagnose it early, or even putting targets for treatment like this. So, thank you.
Yeah, so I mean, I think what Martin is saying there is actually one of the things that I know there's a number of researchers at MBI who are interested in cancer. I mean, Andrew talked about his previous work. And then one of the things that came out a number of years ago was sort of isolating these circulating tumor cells, which are found in cancer, and they can actually, so there's a very small number of them in the bloodstream, but they're a good predictor of sort of diagnosis and sort of future response to treatment. And one of the things that one of the researchers here worked out a number of years ago was that actually these circulating tumor cells are a lot bigger than sort of normal cells in the red blood. So you can actually sort them out just through mechanics. It's almost like a sort of a pachinko machine that you see where the blood gets like poured through something and there's little things to catch them. And they'll specifically catch these larger circulating tumor cells while letting the other blood cells go through. And then with that purified population, you can isolate them and then use them for diagnosis and treatment. And again, so this is a way that you can use just mechanical properties to do early diagnosis for potential treatment. So we have a question here from Isadora, saying that for her undergraduate thesis, she's working on B-cell biology and mechanosensing. And basically, how challenging is it to move into a future PhD or other career in mechanobiology? I'll pass this over to the students, the PhD students in a second. I will say that our graduate program is open up to all STEM students. So we have people on our graduate program, again, with backgrounds in sort of pure mathematics and physics, biomedical engineering, and some with, very much a biological life science background. So we know that with that sort of broad level of students incoming, everyone needs to be brought up to speed so that you can handle. So we've got people from biology who may not have handled sort of large data sets. And on the other side, we have people coming in from maths and physics who've never gone into a wet lab and handled a pipette and done those kind of like lab experiments. So what we actually do have is a sort of initial sort of boot camp session. So is it a 2-week course? About 2-week course to basically refresh the areas that people are strong at and that they're undergraduate in, and teach them about new aspects of basically interdisciplinary research. So the quick answer would be that we're not expecting everyone to come in with a mechanobiology background. You will be fully trained up as part of your PhD, as part of the PhD program for this. Is there any, Jashan or Malia or Martin, is there anything that you'd like to add on that?
I think I can add in a little bit. So you asked how much engineering and maths do you require for mechanobiology. So there are varying level of maths and engineering that you may require. Can be ranging from 0 to all the way to only engineering and maths. So it's actually up to your preference. So if you want to go really theoretical, or if you want to go more biological, so you can choose that varying level of engineering. And saying that you can actually, it's not difficult to train in the techniques, and there are people around you with that varying levels of experience in engineering, and they'll be easier. It's easier to guide new students in all types of engineering fields as well.
Does anyone else?
I think I can also add some comments here. Since my background is pure biology, I think I'm similar situation like the question mentioned. So for myself, like experience, for example, I did a lot of imaging, and which also requires like imaging analysis. But I'm not really a coding person. So I think the best thing in MBI is first we have different like faculties who can help us to do like help you coding or help you to do like the technical issues, help you fix up that. And also like for our community is kind of different multiple like background persons. You can always talk to your colleagues or classmates to find them. So I think you do not need to worry about that much. You can always find help and support in our MBA.
Yeah, so one thing also just to follow up on that is like one of the ways that MBI works that can be a little different to other laboratories and research institutes is that we have a sort of open lab. So a lot of, unlike some other research institutes or labs where sort of a lab is over here and all the people who work there are in this one area, we actually deliberately intermingle staff, so postdocs, students from different labs. So you'll be sitting or, in close proximity to other people with different backgrounds. And part of this is to just promote more discussion, collaboration, and for situations like Jashang and you have said, is like, not every people here have different backgrounds. You may not have all the answers for what you want to do on your project, but by talking around and discussing with the people just close to you, you'll find that like, oh, actually, they have a background in a model organism or in mathematical modeling, which can be applied to your project and advance basically everybody's science in that collaborative manner. So yeah, I hope that answers your question there. And if you're already doing something on, mechanical sensing as your undergraduate, that's already, I think, part of the way there. So yes, we're probably got about another 5 or so minutes. So if there's any more questions, do put them in. I did want to ask our students also about, I guess, like, can you give any interesting experiences either at MBI or at Singapore about living here? Was it what you expected, or was it very different in any ways?
Okay, I can go first. Yeah, so for me, from my experience in Singapore, what I like most about Singapore is the safety. Actually, I was surprised that you can leave your stuff somewhere and you come back and find it without anyone touching it. So I was very surprised about that, because in my country, not bad. In my country, they can even take something from your pocket. So I was very surprised that you can leave something here and then you can, yeah.
So I came during the COVID period. So for the first 1 year of my PhD, it was all lockdowns, and after that one period, it was all open. So it was a Singapore 1 and Singapore 2 for me. So I'm still exploring a lot of Singapore, and I have to say it's quite safe is a very nice word, but it's also quite happening. The nightlife in Singapore is quite nice, and you can roam around the whole island. Pretty much anywhere. So it's a nice place to stay, I would say.
Do we have any other questions from anyone, either in the chat or online? Yep. So how about you, Mengya? Was there anything that's been surprising or thing about MBI or Singapore here?
I think first of all, I think Singapore's culture is very close and similar in China. So, but another thing which is very surprising to me is like a gesture, like Zambaf. So, like in China, we use this gesture to say, "Oh, you're doing great." But I realized in Singapore, so this gesture is like can be used to say thank you, to say like okay. So I realized, oh, it's just very funny thing. Like sometimes you can use one gesture to all like situations like.
Yeah, I think that's one of the things that has been reiterated by Andrew's presentation, how sort of global our community is, but also I think how sort of welcoming and sort of global Singapore is as a place to work and do research. Okay, so I know that we're coming to your end. I guess I'd just like to ask, let me go back to Mengya first. Is like, do you know what you plan to do after your PhD? Do you wish to stay in Singapore in academia or move into something a little bit related?
Yeah, so for the future study, currently I think I really enjoy to do science in Singapore because it's like very cozy, like you can do things, follow your own like to-do list. So for future, I think I would firstly think about stay still in the academia and to do postdocs. Maybe overseas, I'm not really decided at this moment. So yeah, so it's not still an open answer question currently. I'll still like explore that in my future.
And Jasha, I know, like you said, your background, you've actually done some work for various what sort of biotech company or sort of life science. So, is that something that you're thinking of, or have you been sort of focused in an academic career?
I think so. I would like to transition into industry after the PhD, maybe an industrial postdoc or an industrial position. So it's still open up for how the PhD ends. But if I have to stay in Singapore or not, I think for the next couple of years, Singapore, I would stay in Singapore, but then I'll see if, how opportunities present itself.
Have you had any sort of experience or exposure to that sort of translational or, I guess life science, business or tech culture here?
Yes. So there's a lot of companies, I would say, in Singapore that facilitate PhD transitions like SG Innovate, Entrepreneur First, and all of these companies. And here at NUS Grip also, that try to take your science and translate it into a product. So these can be quite helpful for Singapore and for yourself to take your science out of the lab and into the market.
And that's also available for,
Yes,
PhD
yes,
students
yes,
as well.
yes,
It's not just
whoever-
to established researchers.
Yes,
Okay. Okay, I think Martin, I guess you can close us up. I know you're not quite as far along in your PhD as these other two, but yeah, have you had any thoughts yet about what you plan to do afterwards?
Yeah, so my plans are also for now to remain in academia. Yeah, even before coming here, I was working at the university, like teaching, not necessarily research, but more say teaching. But at the moment, I'm thinking of maybe doing the postdoc after my PhD and continue with academia.
Okay, so I know we have another few minutes where everyone's getting ready for the plenary lecture, so I think we'll probably close it off there. I will say, let me just do the last, just reiterate some of what's being said. Is like, if you found this, thank you everyone for listening and attending. And if this has sort of raised your interest into what is this new-ish field of mechanobiology, and you're inspired to learn a bit more, then please come and have a look at our website, bottom there. So mbi.nus.edu.sg, and you put /education or /jobs. You can see what sort of opportunities are available. As mentioned, we have a sort of fully funded undergraduate summer training program that is currently accepting applications until the end of January, where we'll basically take a cohort of undergraduate students. They can be from Singapore or from overseas, and you'll come here for a fully paid 2-month internship to do a research project and learn about mechanobiology. We obviously have our PhD scholarships, which are available. I think we may have just closed a deadline for the next intake, although if you are interested, then do have a look and drop us an email, because I think we can discuss that if there's some last minute applications. And there's definitely a large scope of postdoctoral fellow positions here. And again, if you're further along in your academic career and looking for sort of an independent position, and this is something you're interested in, do have a look at our page there for faculty recruitment at all levels there. So with that, I'd like to thank everyone for listening, and I'd like to thank Andrew, Jashan, Martin, and Mingyu for participating in this. And I hope you enjoy the rest of the GYSS events that are planned for you. Thank you very much.